Hi, everyone. So my name is Marlene Carla. I prefer Carla. Um, so today I'll be talking about the adoption or removal of pharmaceuticals from aqueous solution using biochar derived from cotton gin waste and Wayuli bagasse.
There you go.
Okay. So just a little bit of motivation for my research. It stems from the fact that the WHO estimated that by 2025, more than half of the world's population would be living in water stress conditions. And agriculture that uses most of the water in the United States will be negatively impacted by the scarcity. And so in an attempt to preserve the limited water resources, treated wastewater has increasingly been used for irrigation during farming. However, conventional wastewater treatment plants are not equipped to remove pharmaceuticals from water. And so when this water is being reused, you have the transfer of pharmaceuticals into the soil where they can leach and also contaminate nearby water bodies and groundwater. I don't know why this is. So in an attempt to mitigate this problem, in our research, we proposed the use of biochar. And so biochar is this charcoal-like substance that is produced when you burn biomass at really high temperatures in the absence of oxygen. And so biochar has been produced from several different feedstock, and principally the feedstock used to produce biochar come from waste biomasses. So you have like rice husk, corn stover, and even orange peels. And the reason why biochar has gained so much attention over the years is because not only has it proven its ability to remove several contaminants from water, such as heavy metals, and it can be used to replace activated carbon because right now activated carbon is not a renewable material and it's also obtained either through wood or coal. Also, biochar can be used to increase soil fertility, and it also serves as a waste management tool because most of the biochar is produced from trash that would have otherwise ended up in the landfills. So the main objectives of this research are divided into three sections, if you will. The first one was to just look at the whole biochar production process, especially the temperature, and see how this would affect the resulting biochar that we end up with. Next, we wanted to understand the interactions that might exist between the biochar and the pharmaceuticals that will ultimately lead to biochar being able to remove these pharmaceuticals from aqueous solutions. And we did this by using batch adsorption experiments. And lastly, we also wanted to know how this changing the pH of the solution affects biochar's ability to interact with the pharmaceuticals. So to produce our biochar, we decided to go with waste streams from two plants. So the first waste stream is cotton gin waste, which is produced after, which is kind of like the byproduct that is left after the cotton has been harvested. And the reason why we chose this feedstock is because over 2.5 million metric tons of cotton gin waste is generated annually. The second biomass comes from the Wayuli plant. Yes, it's spelled with a G, but it's pronounced Wayuli. And it's a desert shrub that grows in arid parts of the United States. It's principally used to produce latex. But the problem here is that only about 5 to 10% of the plant goes into latex production, and the remaining is left as waste. And so we had to find a way to transform this trash into treasure. So once we have a biomass, we then decided to produce our biochar, and the process is fairly simple. So we would take our biomass and place it in the reactor and heat it at three different temperatures. Remember, one of my objectives was to look at the effects of temperature, and that's why I chose to do it at three different temperatures in a steady stream of nitrogen gas because this is an anaerobic process. And so at the end, I have 6 biochar samples just because I started with 2 feedstock, pyrolyze it at 3 different temperatures. So if you see me referring to something as GB 700, It just means the Wayuli bagasse that was pyrolyzed at 700 degrees Celsius. And if I say CG500, it's just the cotton gin that was burned at 500 degrees Celsius. So remember, I said I wanted to look at the properties of the biochar and how that varies with the temperature. And one of the properties we're really interested in is the surface area, just because prior studies say that when you have a high surface area, there is a higher ability for biochar to remove more of the contaminant that you have in solution. So something that's very common with both of our biochars was that at 700 degrees Celsius, we had the highest surface area, which is what we would expect, because when you increase the temperature, you have a release of organic matter that was either inside or blocking the pores. However, our surface area was still really low when you compare our surface area to the surface area of activated carbon that can go up to about 300 meters square per grams. Our surface area is fairly insignificant, but we still decided to test the biochar. Another property we decided to look at was the pH, because through this, we can hypothesize whether our biochars are hydrophobic. So once you take a little sample of the biochar and mix it with DI water that's typically at a pH of around 7, and you notice that your the mixture increases in pH dramatically, that means you have a fairly hydrophobic compound in your hand. So our biochar is fairly hydrophobic. And so we decided to look at biochar's ability to remove 3 different pharmaceuticals. You have sulfapyridine and erythromycin that are both antibiotics, and you have Docazide that is used as a stool softener. And the way we chose our pharmaceuticals was we looked at the pKa, which is the acid dissociation constant, and all of them have varying degrees of pKas. And we also looked at the log KOW value, which will tell us the likeliness that our pharmaceuticals have for water. And so this is the setup for our batch adsorption experiments. It was fairly simple. So what we did is we took some DI water and added a known concentrations of our pharmaceuticals to that water. We started with an initial concentration of 10 milligrams per liter. And then we would take each of our biochar samples, 0.5 grams of that, and mix it with 100 milliliter of the contaminated solution. We will shake this setup for about 24 hours. And during that 24-hour time period, was sampling in between. The samples were filtered and then sent out for analysis. We also covered the bottles with aluminum foil just to make sure that we account for any removal due to photodegradation. So now I'm going to talk a little bit about the results. I'm going to talk about it generally, about some of the trends we noticed. But before I do, I want to mention that biochar's ability to remove pharmaceutical comes from an interplay of different mechanisms, such as electrostatic attraction. You have hydrophobic partitioning. You have the formation of hydrogen bonds between the biochar and the pharmaceuticals. And you also have what is called high-pi electron donor interactions that can occur between the pharmaceuticals and the biochar. And also, the results were reported in terms of removal efficiency, where if you have 100% removal, it means you were able to remove all of the pharmaceuticals that were placed in solution with the biochar. So now these are the results for the sulfapyridine. Remember, this was an antibiotic. On the y-axis, you have the percentage removal, and on the x-axis, you have the 6 different biochar samples that were tested. So not all of the biochars that were in solution were able to remove sulfapyridine, first of all, but something that we noticed was that surface area really played a role in the removal of sulfapyridine, as the cotton gin 700, which was our biochar with the highest surface area, also had the highest removal. Another thing that we noticed played a significant role in removal is the log KOW value. So sulfapyridine has a log KOW value of about 0.35. So it's it's fairly hydrophilic, and so it would rather stay in solution than partition into the pores of the biochar. Another mechanism that was responsible for sulfopyridine removal is the ability for sulfopyridine and biochar to form negative charge assisted hydrogen bonds. This is because at the pH, the solution pH, which was around 10 to 11, sulfopyridine is negatively charged and our biochar is negatively charged. And together, they were able to form those hydrogen bonds due to their negative charges. Next, you have the removal of docosahexaene, which is our stool softener. And you can see here that docosahexaene was removed by all of the studied biochar with, again, the cotton gin that was pyrolyzed at 700 degrees Celsius, showing the highest removal, principally due to the fact that it was our biochar with the highest surface area. Also, the main mechanism responsible for docosahexaene, for the removal of docosahexaene, was the fact that docosahexaene can partition hydrophobically into the biochar. And this is because when you look at its log KLW value, it's 5.24. It had the highest log KLW of all of our compounds we decided to test. And it really shows because all of our biochars were really able to remove docosahexaene. So it would rather partition to the biochar instead of staying in solution.
3 minutes remaining, Carla.
Sure. So now you have the removal of erythromycin. Also, erythromycin was removed by all of our biochars. And this is also because it has a fairly high KLW, and but its KLW is lower than docosahexaene. So that's one of the reasons why it wasn't removed to the same level as docosahexaene. Also, if you look at the structure of erythromycin, it has all these OH groups, and so it's able to form hydrogen bonds with the biochar. So next, we decided to test the effects of pH. And the way we did this is we took the cotton gin 700, which was our biochar with the highest removal efficiency. We placed it in solution with the compounds we're trying to remove, and we decreased the pH of that solution to about 7 using nitric acid. And so we decided, we still ran the experiment for about 24 hours. And you can see that as you move from pH 10 to 7, the removal of sulfopyridine decreases. And this is because sulfopyridine was mainly removed by the formation of negative charge assisted hydrogen bonds, because at a pH of 10, it's negative. But when you bring down the pH to about 7, it's mainly neutral. And so the formation of those hydrogen bonds no longer exist at a pH of 7. And this is why the removal decreased. Here you have the removal of erythromycin and docosahexaene. We didn't really see a significant difference with the removal at 10 versus 7. And this is principally because the mechanisms that were responsible for the removal of these compounds, which are hydrophobic partitioning and hydrogen bond, existed throughout the entire pH range. So just to summarize our findings, 1, we noticed that the mechanisms involved for the removal of pharmaceuticals consisted of hydrophobic partitioning, hydrogen bonding, and the formation of negative charge assisted hydrogen bonds. The highest removal of pharmaceuticals was using the CG700, which had the highest surface area. The addition of sulfopyridine decreased from pH 7 to 10 due to a decrease in the formation of negative charge assisted hydrogen bonds. And the results suggest that biochars from cotton gin waste and Waioli bagasse could act as effective adsorbents to remove pharmaceuticals from water. So these are just the people I'd like to acknowledge. And thank you.
Great. Thank you so much. That was a great presentation.
Thank you.
Yeah, I think we do have about a minute for questions.
Sure. We have a question about whether the biochar was activated or if it was regular biochar.
It was regular biochar, so it was just pyrolyzed and we didn't do any chemical treatment to it.
And one more about, did you do any reversibility studies?
Oh, that's what we're planning on doing right now, because it's fairly hard to do it with batch adsorption experiments. So we're trying to do it with filtration experiments. So right now we're moving towards filtration experiment and we're going to test the reversibility.
Sarah, do we have time for one more?
Yeah, let's do one more.
Did you do competitiveness towards binding study sites?
Yeah, well,
site study.
Not for this study, but right now that's what we're doing. So right now we're moving towards doing a mixture of pharmaceuticals, and we're also including organic matter into the water and see how all of those contaminants compete for the absorption sites.